Stem cell therapy has rapidly emerged as one of the most promising and ambitious approaches to reversing the cellular markers of aging, particularly through its potential influence on telomere length and overall genomic integrity. At the heart of this concept is the remarkable ability of young stem cells, especially pluripotent or multipotent types, to regenerate tissues and restore cellular function where degeneration has occurred. These stem cells naturally possess long telomeres and active telomerase, meaning they are biologically younger and more resilient compared to senescent or terminally differentiated cells in aged tissues. This unique feature makes them attractive candidates not only for regenerative medicine but for strategies specifically aimed at rejuvenating the aging process at the molecular level. Telomeres, as protective caps at the ends of chromosomes, shorten with every cellular division. In most adult cells, telomerase is inactive, so each replication brings the cell closer to senescence. Stem cells, in contrast, maintain active telomerase expression, allowing them to preserve telomere length and continue dividing over long periods without degradation. By introducing these cells into aged tissues, researchers hypothesize that it's possible to reset the biological age of those tissues, at least locally, by replacing old, telomere-depleted cells with youthful, robust ones. Studies in animal models have supported this idea. For instance, transplanting young stem cells into aged mice has led to improvements in organ function, increased muscle mass and regeneration, better cognitive performance, and even extended lifespan in some cases. The mechanism goes beyond mere cell replacement. It also involves the secretion of beneficial signaling molecules known as the stem cell secretome. This collection of cytokines, growth factors, and exosomes can modulate the local environment, reduce inflammation, promote tissue repair, and possibly induce rejuvenating effects even in neighboring aged cells, an effect sometimes called paracrine rejuvenation. Moreover, stem cell therapy is being actively explored in combination with other telomere-preserving strategies. For instance, induced pluripotent stem cells, iPSCs, adult cells reprogrammed back into a pluripotent state, have been shown to restore telomere length during the reprogramming process. These cells, once redifferentiated into target tissues, could theoretically be used to repopulate organs with biologically younger cells. Recent experiments even suggest that partial reprogramming techniques can reverse aging phenotypes without erasing cell identity, offering a potential future where cells can be rejuvenated in place rather than transplanted. Yet despite the optimism, major challenges remain. One of the most significant is immune rejection. Unless the stem cells are autologous, derived from the patient's own body, they may be recognized as foreign and attacked by the immune system. This not only reduces efficacy, but introduces serious safety risks. Even autologous iPSCs carry risks of mutation during the reprogramming or differentiation phases, raising concerns about tumorigenicity. Teratoma formation, benign tumors consisting of multiple tissue types, is a known risk with undifferentiated pluripotent cells, making precise control over cell fate critical before clinical use. Another major obstacle is integration. For stem cells to meaningfully improve tissue function, they must not only survive transplantation but differentiate appropriately, integrate with host tissue, and perform the needed physiological roles long-term. This is especially difficult in complex organs like the brain or heart, where architectural and functional precision is paramount. Despite progress, achieving full functional integration without adverse effects remains a scientific and engineering challenge. Nevertheless, progress is accelerating. Clinical trials are underway for mesenchymal stem cell, MSC, therapies in conditions like osteoarthritis, heart failure, and neurodegenerative diseases, all of which are linked to age-related tissue degeneration and, indirectly, to telomere attrition. While these treatments are not yet focused solely on telomere length, the systemic improvements they produce may reflect underlying enhancements in cellular vitality, reduced inflammation, and restoration of regenerative capacity, all hallmarks of slower biological aging. One especially exciting frontier is the potential use of gene-edited or enhanced stem cells. By combining stem cell therapy with technologies like CRISPR or epigenetic reprogramming, researchers could theoretically produce cells that not only retain youthful function but are also resistant to oxidative stress, senescence, or telomere erosion. This opens the possibility of producing next-generation stem cells with optimized longevity and safety profiles, designed specifically for anti-aging interventions. In conclusion, stem cell therapy offers a powerful, multidimensional approach to combating aging at its roots. While technical and ethical challenges persist, particularly around immune compatibility, safety, and regulatory oversight, the ability to repopulate aging tissues with telomere-rich, regenerative cells is among the most biologically plausible ways to restore function and delay the onset of degenerative disease. As techniques improve and safety becomes more reliable, stem cell therapy may become not only a tool for treating specific conditions, but a cornerstone of personalized strategies to slow, halt, or even reverse aspects of biological aging itself.